Hello, everybody. My name is Konstantinos Mikmizos. I'm an assistant professor of computer science at Rutgers University. And this talk will present a snapshot of our efforts in pushing the envelope of neuromorphic computing. I'll provide hopefully measurable evidence that adds value to this new emerging and fascinating field. So by the end of this talk, you will hopefully know a few more things on how intelligence emerges in the brain and how we're actually using this new knowledge to design brain optimized solutions. I'll start by giving the main message right up front and say that the brain has been computing long before we started studying it. Brain studies have been really slow in informing models of brain computation. For example, the computing principles that convolutional neural nets or capsule nets are using are actually derived from biological mechanisms discovered in the cortex in the 1950s. It is also worth mentioning that the explosion of neuroscientific knowledge over the last 3 decades is still mainly out of reach for most AI solutions, partly because this knowledge is not accessible by the technical AI people, and because these mainstream AI formalisms such as backpropagation are not flexible enough for this new knowledge to be incorporated. For example, and this is where this talk is focusing today, over the last century, the brain has been studied through recordings of its electrical activity. And this has created a dogma, a conception, a misconception that brain computation equals neuronal processing. And this dogma has innervated most hypotheses on how the brain works and has also innervated our efforts to translate this knowledge into models of artificial intelligence. Even neuromorphic computing has inherited the fundamental limitations that AI has, including the so-called neuron doctrine, essentially that the neuron is the only processing unit in the brain, something that is now arguably known to be incorrect. Today, we know that other non-neuronal cells, named astrocytes, play a key role in processing and learning information. Strikingly, there are not enough efforts to translate their computational roles into artificial networks. So our fundamental theoretical approach is that brain-inspired computing can scale by further pursuing its biological plausibility. To achieve this goal, we computationally capture recent breakthroughs in our understanding of how intelligence emerges in the brain. We then translate these cellular principles of intelligence into learning algorithms and brain-optimized architecture that span a range of brain medices. To evaluate our theoretical approach, we harness the emerging human-like behavior into neuromorphic hardware, and we benchmark our brain-optimized systems in solving foundational problems in robotics. So over the last few years, we've given evidence that the neuromorphic solutions to real-world problems are robust, they're versatile, and they're orders of magnitude more energy efficient when compared to the mainstream solutions, including deep learning. Turning our attention to astrocytes, experiments at the behavioral level have showed that transplanting human astrocytes in mice improve their cognitive functions as well as memory. Recently, it was also found that astrocytes are not only necessary but also sufficient for forming new memories in the brain. And even at the synaptic level, astrocytes change the synaptic weights with a mechanism that deviates from what we know about, for example, local synaptic routes such as STD. So these are the main reasons why we examine the computational roles of astrocytes in learning and memory. We steered our research from the technical point of view to Hopfield networks, and we did that for two reasons. Hopfield nets are the precursors of modern networks used in machine learning, say, for example, Boltzmann machines or deep belief networks. And from a neuroscience perspective, Hopfield nets are models that represent memories in the form of energy minima that are stored in the synapses, where astrocytes and neurons are arguably interactive. Very briefly, a Hopfield net is a recursive, fully connected network of binary state neurons, where memories are stored as values of a global energy function that is associated with the state of the network. So each neuron processes locally the decision whether it should flip its state or not, and this causes the overall network to be attracted into a local minimum, which is actually one of the memories that have been stored. So our goal with the Hopfield net is to add astrocytes and see how they can generate a transition between memories at the neuronal component of the network. So to do so, we introduced, as you will see, a different type of learning, an astrocytic learning, to modulate the neuronal component of the network. Now, sequences of memories are widely seen in biology in cases such as, for example, walking, chewing, or breathing, and the ability to store sequences of memories is related to our ability to actually learn new tasks. So I'll describe how we explored and exploited the computing principles of the astrocytes as have been identified in experimental studies by deriving the equations that I'll describe in order to push a Hopfield net from one memory to the other. Everything that you see as black fonts is old news. It's something that has been developed over the years. With the green fonts, I have marked our own contributions as described in our paper. So we started with n binary neurons with symmetric weights, and the field of each neuron is actually the result of two components, the neuronal component and the green one, the astrocytic component. So we focused on the ability of the astrocytes to modulate the neuronal activity using the so-called slow injected current, or SC, essentially a current that they introduce into the postsynaptic neuron. To model that current, we had to derive the dynamics of the calcium wave increase in response to the presynaptic activity, as well as in response to the dynamics of the injected current. So we derived the time needed to be spent on each memory separately. And we did that by solving the simple differential equation that you see here. We found that the time is actually a function of the threshold limit of the calcium as it increases, as well as two parameters, alpha and beta, that essentially determine how much we rely on the previous calcium concentrations to estimate the next ones, as opposed to the contribution that is directly coming from the presynaptic neuron. So we made this assumption that, in fact, the two parameters alpha and beta should be related. So we derived the time tau that we spent on each memory, and it's better actually to visualize it as a function of alpha, beta, and the threshold for the calcium. So this plot shows that the more confident an astrocyte is, the more higher the threshold should be, and the more time it takes for that transition to happen. So if you think about it, this is exactly what is happening when we learn. As we learn something and increase our confidence, we tend to become faster and faster in doing that task. I'm just mentioning a small detail here. The slight divergence in the assay timing that you see in the beginning is due to the asynchronous dynamics of the neural network during the state transition. And I'm going to give you now the intuition behind this mathematical loaded slides, is that essentially the network relaxes into a local minimum, into a memory, and by the time that this happens, the astrocyte starts to kill the energy field into the next memory, and then to the next one, and so on and so forth. The astrocytes learn to do that, at least in our world, with that simple Hebbian-type astrocytic learn that incorporated a learning rate, as most learning rules, as well as the time between switching between memories. That was the most important thing. We needed to show to the astrocyte enough of that specific memory so that it can capture it in the calcium activity. It also relied on the postsynaptic state as well as the astrocytic state that over time matched the presynaptic state. So overall, our contributions is that we showed how astrocytes can learn to change memories, to trigger state transitions in a Hopfield-type associative network that can be used to model memories. We derived the dynamics of the astrocytic activity by synthesizing math and experimental studies. And our overarching goal is to use this type of research and introduce astrocytes with their new neuromorphic and neuromodulatory roles into neuromorphic chips, such as Intel's Loihi, essentially introducing a second processing unit to neuromorphic chips. That's all I wanted to say. I hope you are all staying well and staying safe, and I really hope we will meet each other again soon.